This is Israeli Technology Founders Speak, a podcast of conversations with successful Israeli high-tech and biotech entrepreneurs. With your host, Avraham Hermon. Racheli Ofir is the CEO and CTO of Betalin Therapeutics, an Israeli company developing a biological micropancreas that could one day offer a one-time cure for insulin-dependent diabetes, replacing the missing pancreatic function with living cells that sense glucose and respond in real time. Avraham sat down with Racheli to discuss how the technology works, what their animal trials showed, why IP is critical, the challenges of funding a biotech startup in wartime Israel, her advice for startup founders, and much more. This podcast is a creation of J.M.B. Davis Ben David, an intellectual property law firm serving clients around the world. You have great innovations. We keep them safe. It's not enough to just have a great startup idea or innovation. If you don't legally protect your innovations, products, and brand, anyone can claim them as their own. We keep your great innovation secure. Learn more by going to jmbdavis.com. That's jmbdavis.com.
I'm here with Racheli Ofir, CEO and CTO of Betalin Therapeutics. First, let's start with what problem Betalin solves.
So Betalin is aiming to solve the problem of diabetes, which is a huge problem, especially insulin-dependent diabetes. There are over 5 or even 600 million people suffering from diabetes in the world, and a large part of them are insulin-dependent. And right now they are dependent on the combination of devices, a device that measures glucose levels in the blood of the patients, and then another device that corrects the glucose levels by injecting insulin. They are really sophisticated devices, but still it's a huge burden on the quality of life of these patients. They need to monitor their glucose every few hours, before doing sports, after doing sports, before eating, after eating, etc. And usually they don't reach full normal glycemic values. And therefore we want to solve the problem from the core, which is the lack of a functional pancreas. And this is by injecting or transplanting actually a functional micropancreas. And this is the technology that we're developing. We're building a biological pancreas. It's not like some people call a closed system devices, they call them artificial pancreas, but we're talking about an actual biological pancreas, which is composed of biological tissue with insulin-secreting cells embedded in it. And together, it's a whole, fully functional organ that can be transplanted into the diabetic patients. It can sense the glucose levels in the blood of the patients and provide the right response, the right level of insulin that is needed at that particular moment. The response is by far faster than the response of a device, which takes a few seconds, if not minutes, and takes it time. When we're talking about a biological tissue, everything happens within microseconds, so there are no times of imbalance in the blood glucose levels.
Okay, so the times in the blood glucose levels are small, is what you're saying.
Yeah, because it's a biological, the time of sensing and responding is by far faster when we're talking about a biological tissue if you compare it to a device.
This sounds almost like science fiction. How did this idea come into being?
So the idea originated from the Hebrew University, and the idea was that cells need their microenvironment. The cells in the body, they're not just floating around, they're embedded into tissue. The tissue is not just a physical anchor for the cells, but it also the way that signals pass between the cells and the tissue, and the cells need to interact with each other and with the tissue. So the initial idea came from the Hebrew University, from Professor Eduardo Miterani, and it was taken by Betalin and developed much further into where it is today. To use real biological tissue And in our case, we're using porcine lungs as the source of the tissue that we're using to implant the pancreatic cells on them. And why porcine and why lungs? One, porcine pig tissue is very similar in the biological composition, in the proteins, to human tissue. We're quite close, and as pig tissue is used for heart surgeries and other medical uses and is known to be biocompatible for human. Why lungs? Because lung tissue is like a sponge. It has a very large surface area. It's built of many alveoli, which provide a very large surface area, and we can implant very large number of cells on relatively small tissue. This structure provides protection to the cells and the right microenvironment for them to succeed. There have been attempts by other companies in the past to implant pancreatic cells, islets from cadaveric donors, or even stem cells into people. And the bottleneck is how to make it succeed, how to enable the cells to function and survive for a very long time. This is what we expect them to do. And it was found that without the supportive tissue, this cannot succeed. The cells need this microenvironment to support their survival and long-term functionality.
So you're using what sort of pancreatic cells?
So we started with a cadaveric-derived donor-derived islets, and now we are using stem cell-derived beta cells. So these are the sources, embryonic stem cells that have been differentiated into beta cells. We are not the developers of the cells. We work with several other companies that are developing the cells, and they are also aware of the need for the supportive tissue. And this is the bottleneck of the entire industry, how to successfully implant the cells.
I see. So you're using a combination of porcine cells with stem cells together in your unique model to...
Not porcine cells. Actually, our technology is very much related to the removal of the porcine cells from the porcine tissue. So we want the extracellular matrix of the porcine. We want the biological tissue, but we definitely don't want the porcine cells. And this is a little bit tricky because you want to get rid of the porcine cells, and using enzymatic proteins, for example, you would remove the cells but also harm the tissue, which is also made of protein. So
I see.
We need to remove them without affecting the biological structure and the physical structure of the tissue.
I see. Okay. So how have you shown that this can work? What sort of testing have you done so far?
So we started, as every development of a medication, of a biological product, we started with in vitro testing, and we saw that the cells survive much longer on the scaffold than freely in the medium. We saw that seeding them on the scaffold, which is what we are developing, enables them to survive much longer even in vitro in a Petri dish. And very importantly, they provide them the ability to respond to changing glucose levels, which didn't happen when the cells were grown without the scaffold. And then we moved into diabetic mouse model, and we transplanted the micropancreas, a combination of the scaffold with the pancreatic cells, the beta cells. And we saw that indeed we saw secretion of the human insulin from the cells in the blood of the mice, and very importantly, regulation of the blood glucose levels of these diabetic mice. And very importantly, the micropancreas survived for several months in the bodies of these mice. And when we removed them out, we saw that there was vascularization of the mouse, connecting the transplanted micropancreas with the body of the mice. We were very pleased to see that, but it's not that surprising since we saw that the insulin secreted from the scaffold is found in the blood of the mice. And because we saw that the glucose levels were regulated, it made sense that there was a vascularization, but seeing that after removing them made us really happy.
So I guess the next stage is testing it in humans?
Yeah. This is what we're aiming to do. This is the next stage.
Okay. And how often would you have to give this sort of treatment? You said that it lasts for a number of months in mice.
So the vision is that it will be a one-time treatment.
Wow.
But We will need to prove it in human that it works and maybe improve it based on the findings in human. But this is the vision that it will be a one-time treatment. And it's actually changing the way that we are looking at diabetic patients. Now these patients are managing their disease, but we want to cure them. They don't have a functional pancreas, and we want to provide them with one.
So let's talk a little bit about your investment. You had mentioned the technology comes from the Hebrew University. Where does the investment come from?
The founder of the company, Shuki Ershkovitz, he was able to raise some private investors, which started the company. We were very fortunate to receive funds from research grants, both from the Horizon 2020. We received 2 grants from them, and also from the Israeli Innovation Authority. This is the funding that we have received so far.
I see. And let's talk a little bit about intellectual property. How important is intellectual property, such as patents, for your company and for the investors?
It's critical. It's critical both for us and both for the investors. One of the first questions that is raised by potential investors is if we have IP, if we have granted or pending patents, in which countries do we have IP? And of course, it's important. As I said, it's a huge market, and once this grows, of course, we will need to be able to stop others from doing what we are doing. So yes, it's critical. We have 3 patent families, and one family is already granted in most countries with continuation applications, and we keep filing applications as we move forward with the development.
Okay. So you're located here in Jerusalem. I was just commenting on the way in about how beautiful the view is here from the Ein Karam area of Jerusalem. And I guess it's Israeli technology from Jerusalem. You're working in Jerusalem. What impact does being in Israel, in particular Jerusalem, have on Betalin?
So first of all, we're very proud to be an Israeli Jerusalem-based company, and that the technology came from Israel. There are some benefits for Jerusalem-based companies, but these days there are also hurdles related to being an Israeli company in terms of raising funds. People are worried. This is a war zone. They're more concerned about investing in Israel at these times. But
what do you say to such investors that are concerned? What's your response to them?
What do I say? We believe in our technology. Israel has been struggling for the past almost 80 years, and we are here, and the technology will be here. I really hope to grow to be an Israeli company. I don't want it to have a spin-off to a US company. Sometimes you have to follow the money, but I really think the technology grew here. The people who developed it and invested in it, the private investors, most of them, not all of them, are Israelis. I think this deserves to be further developed in Israel.
Okay. Let's talk a little bit about your hurdles moving forward as a company. What sort of hurdles have you had and overcome?
So, of course, developing a drug, a biological drug, is a challenge by its own. There are the technological challenges, the regulatory challenges. It's challenging. And on top of that, there's, of course, the financial challenge of funding this. Large VCs or big investors, they're waiting for you to be further down the road. And to be further down the road, you need funding. So it's a lot.
Chicken and egg problem.
Yeah.
So let's talk a little bit about challenges that you've had, or perhaps even opportunities that you've had, based on being a woman who is involved in the company as CEO and CTO.
Interesting question. So in theory, I would like to say that there are no challenges of being a woman, and everybody is based on what they do. I think it's more challenging in the financial world. In the technological world, I think it's more common that women are leading research and development and drugs. I think the financial world is more of a man's world, but I do it in my way, so I think it's okay. I don't think it changes anything, but it's another language and maybe different kind of people than the people that you come across in the technological world.
I see. Can you talk a little bit about the team? who's on the team and how the team was formed.
So the team is based on mainly scientists, so almost only scientists except for a few more administrative people. Most of them, or even all of them, live and studied in Jerusalem. So it's a really Jerusalem-based company. People from the Hebrew University, from Michlelet Hadassah, and masters and PhD in biology. Good people.
Okay, good. That's important.
Yeah.
So what tips do you have for founders who are just starting with their new idea or their new company?
Be strong. You need to have patience. You need to be persistent. You need to be patient to cross hurdles. Nobody will come to you and say, oh, this is how you do it. It's really easy. There's no recipe for how to develop it. You have to find your own way. There's no recipe on how to do the regulatory process. Raising money is difficult. I think the number of research grants that we had to submit in order to receive 2 or 3 grants, it's not a very good percentage of success. But once you receive a big research grant, then it enables you to move forward. And grants are amazing because it's also non-dilutive funds. So
Sure.
It's the best kind of money.
I see. Okay. So I guess your recommendations are be patient, be persistent, and also look for the right sort of grants, right?
Yeah.
Okay. Where do you see Betalin down the road, like another year or another 5 years from now?
So in the coming year, maybe a year and a half, we expect to be in clinical trials in human. And this is a completely new game. Playing with mice is great. You have to do it. This is the path. But mice are not little people and people are not big mice. We want a medication for human. We have to enter the clinical trials. This will give us a lot of information on how to move forward, and also in terms of how the world looks at us. A preclinical stage company is completely different than a clinical stage in terms of valuation, in terms of interest of big pharma. It puts us in a completely different place. And this is where we hope and plan to be in the coming 18 months. And in 5 years, I hope we have significant phase 2 clinical data, which shows both the safety and efficacy of the cells. And it's a game changer. Once you have efficacy, clinical data, then you're on the road.
Okay. So this sounds good. It sounds like there are a lot of people that are looking forward to such a solution, which could be easier and improve quality of life for patients that are currently treated for diabetes. So wishing you and your team a lot of success, and thank you very much for participating.
Thank you so much. Thank you. It was a pleasure.
That was Racheli Ofir, the CEO and CTO of Betalin Therapeutics. We hope you enjoyed this episode. There are many more to come. Do you have a great innovation or startup idea? We'd love to hear from you. You can reach out to us by going to our website, jmbdavis.com. And if you go to jmbdavis.com/startup, you'll see we have a special site specifically made for startups to help startups protect their innovations. Please be in touch with us and find out how we can help you. Thank you for listening, and we look forward to bringing you the next episode.